Two of the ISB Microbiome course. My name is Nick. I'm a fourth-year graduate student in the Gibbons Lab, and I'm pleased to be your instructor for today's portion of the course. I'll go ahead and share my slides. Great. So the focus of today's portion of the course is on personalized predictions of microbiome-mediated responses using metabolic modeling. So before we jump into the content today, let's make sure you're all set up with the slides. You can open these slides that we're looking at here using the URL shown. The link should also be in the email that you received prior to the course. And I think the TAs have already posted this link in the Slack channel, so you should be able to find it there. If you have any trouble, just ask in the Slack channel and the TAs will be happy to help you. Quick reminder before we jump in, as with yesterday, this course will be taught across 3 separate modules. The presentation slides that we're currently looking at will contain explanations of the logic and the methods that we're using in the course, as well as links to pertinent materials related to the course today. All the analysis and modeling that we'll be doing, as well as building visualizations of our results, will take place in the course notebook, which is hosted in Google Colab. You can navigate to the notebook using the link on the next slide, but I believe that link is also available in the email that was sent to you, and I believe the TAs have posted that in the Slack channel as well. So you should be able to find it through one of those methods. And finally, as with yesterday, the Slack chat is available to you at any time during the course. If you have any questions or comments, if you have a question, it's likely that someone else probably has the same question. So please don't hesitate to post there, and the TAs will be happy to help you out just as soon as they can. Cool. So before we jump into the slides and talking about some of the methods we'll be doing, let's follow this link here to the notebook and familiarize ourselves there and orient ourselves with the notebook a little bit. So if we follow that link, we'll see this links us to our Google Colab notebook. This looks very similar to the one that you were using yesterday with Christian, but again, the focus of this part of the course will be on modeling microbiota-wide metabolism using Mycom. So let's orient ourselves a little bit. On the left-hand side, we have our table of contents, which will show you through all the steps that we're using today in order to build and model our communities. We also have our file explorer right here, which will show all the materials that we're using in the course shortly. So before we jump into actually building our models, we'll need to set up the notebook so that we can use it for some of our analyses here. So to do that, we'll need to install some packages that we'll be using for our modeling methods here. A quick disclaimer before we get into the setup process. Mycom is the package that we're using here in order to build our metabolic models of our metabolic models of our microbial communities. And Mycom has to do a lot of pretty large and complex linear and quadratic programming problems. In order to solve those, Mycom requires the use of a quadratic programming solver. There's a lot of really great commercial solvers that are pretty expensive, although they often offer free academic use licenses. But for today's course, we'll be using an open source solver called OSQP, which is installed alongside Mycom. OSQP is a great quadratic programming tool that works really well for quadratic programming problems, but it's not quite as accurate for linear problems. If you're interested in using Mycom for your own research, I highly suggest that you look into obtaining a license for another commercial solver like IBM's Cplex or Gurobi, which do offer free academic licenses, as I mentioned. Great, so now that that's out of the way, we can start with setting up our notebook. The first thing we'll do is we will clone the GitHub repository, and this will contain all of the materials that we'll be using for today's course. So let's go ahead and run this cell. Excuse me, I missed a step here. Before you jump into running the notebook, you'll probably want to save a copy for yourself in Drive. So if you go to File and save a copy in Drive, this will open a new copy of this notebook in your Google Drive. And you can name this something, whatever you like. Great. So this is the same notebook, but now it's just your own copy. And we'll be able to run all the same prompts as previously. Great. So the first step, again, is to clone our GitHub repository. And this will copy all the materials that we need for today's course from GitHub into our file explorer. So let's run this cell. And this should go pretty quickly. So this contains all of the materials we'll be using, including the QIIME artifacts that were built in yesterday's part of the course that contain our taxonomic abundance and the taxonomic mapping that we conducted in yesterday's part of the course. Great, so now that that's done, we can move on into installing Mycom. Installing Mycom is pretty straightforward in Python. We can use pip to install Mycom. And again, this will go ahead and install OSQP, our quadratic programming solver, alongside it. You'll notice that this cell has this exclamation point again in front of our command. And that's just telling the notebook that it wants to run this cell as a command line prompt rather than Python. So this should go pretty quickly. This will install Mycom along with several other packages we need and OSQP. Great, so that's done. And now the last step that we need to run in order to set up our notebook is to install a couple other QIIME2 interactions. So like I mentioned, we'll be using the QIIME2 artifacts that we built in yesterday's part of the course in order to build our metabolic models within Mycom. In order for Mycom to be able to interact with those QIIME2 artifacts, we'll need to install these two packages as well. And now this should go pretty quickly. But while that's running, we'll switch back over to our slides and we should be able to see, well, we'll investigate some of the methods and processes that we'll be using today. Great. So yesterday, if you recall, we used QIIME2 to analyze sequencing data, starting from raw data and then using that to examine diversity metrics and map to taxonomy, among some of the other methods that Christian introduced. Today, we'll be looking rather at functional analyses of the gut microbiome. That is to say that rather than looking at what populates the microbiome, we'll be determining what is happening in the microbiome, metabolically speaking, which is not always fully explained by taxonomy alone. So when I first say functional analysis, a few things might jump to mind, like some of the omics methods that are listed here, metagenomics, metatranscriptomics, for instance. Most commonly, to determine some of the function of the microbiome, one might do a metagenomic study, from which you can see what genes are present in the community, map those to reactions, and determine what sort of pathways might be active in the community that's being modeled. There is a caveat, though, that in gene abundance data doesn't always tell us the full story that we're looking for. And so that's because gene abundances really only report to us the metabolic capacity of a community. If you imagine a microbial community with really high abundance of starch degradation genes, you might then infer that a lot of starch degradation pathways are active in that community. But if the host doesn't eat starch, then this is clearly this inference is incorrect. So gene abundances tell us what could happen, not necessarily what is happening. And in the case of functional analyses, we really are interested in what's actually happening biologically in the community that's being modeled. So one school of thought that we'd like to present here is that gene abundances are really cool and we can learn a lot from them, but they don't necessarily tell us the whole story that we're after. So today we're going to explore an alternative approach that one can use in order to analyze the function of the microbiome, which is predicting metabolic flux. And fluxes actually will tell us more about the actual metabolic behavior of the microbiome than abundances are able to. Great. So let's talk a little bit about fluxes, what they are and why we care about them. So rather than abundances, which are a measure of concentration, fluxes are a measure of mass conversion into a system and through internal reactions within that system. Since they represent a rate rather than a concentration, fluxes are typically measured in units of concentration per time, for instance, millimoles per hour. In bacterial communities like the ones we're modeling today, we typically also scale flux by relative abundance in order to account for differential metabolic contributions by more or less abundant taxa in the community. All right, so why do I claim that fluxes are more informative than abundances? Well, if we look at the example on the right-hand side, we see a cell, we see glucose going into glycolysis and then into the TCA cycle. And we see that the cell in question here is importing a lot of glucose. But that glucose is almost immediately being converted into pyruvate as it enters the TCA cycle. If we were to measure the internal glucose levels of the cell, we might see that they're actually super low. And if this was the only measurement that we're taking, this might lead us to incorrectly assume that glucose is not important for the growth of the cell, which obviously we know is not the case, because we can see that a lot of glucose is important in order for the cell to grow. This might seem like a pretty naive assumption, but if you imagine all of the different fluxes occurring in a metabolic community, you can see why looking at fluxes might be more informative than looking at abundances. They tell you not only which pathways are active, but also which precursors are important in order to activate pathways later on. There is, of course, a problem with measuring fluxes, as there always is. And that problem is that they can be very costly and laborious to measure. One thing we could do is longitudinal metabolomics, wherein we take metabolic abundance measurements at consecutive time points, and then use those measurements in order to calculate a rate of conversion between the time points. But this takes a lot of effort, is fairly expensive, and is only limited to those metabolites that are included in our metabolomic panel. Another option is isotopic labeling, wherein we feed a community, for instance, glucose with isotopically labeled carbon, and then we measure how much of the resulting pyruvate includes those isotopically labeled carbons. From that, we can then infer a flux that's occurring in the community. But again, this is even more costly than longitudinal metabolomics, and is still limited to just those metabolites that are being targeted in our panel that we're conducting. So really what we need is a way to easily and accurately estimate fluxes without the labor and expense associated with these methods I've just presented here. Luckily for us, there is already a computational tool that does that. So to do just that, we can use a method called flux balance analysis, which has become more and more popular in microbiome research in the previous years. Flux balance analysis is a computational method that uses genomic information, like the information that we got out of CHIME yesterday, to infer the most likely set of fluxes in a biological system. We can use this to predict the modeling behavior of a microbe using genome scale metabolic modeling. So let's take a look at how flux balance analysis works, some of the assumptions it makes, and how we can use this tool in order to infer fluxes in our system that we're modeling. So like I mentioned, FBA is a powerful computational tool that allows us to infer fluxes through reactions in a system by reducing the possible solution space of our fluxes to one that's biologically relevant. So that means that it's a solution that fits what's actually happening in the system that we're measuring in vitro. FBA makes a couple of assumptions, the most critical being that the system being modeled, in this case, the metabolism of a single microbe, is in a state of constant flow or steady state. This just means that the inflow and the outflow of reactions are balanced. A good analogy to this is imagine opening a window in your house. You'll have air flowing in and air flowing out, but you won't suddenly have more air flowing in than out. Suddenly your house expands and pops like a balloon because the air pressure in your house has suddenly increased. That doesn't happen. So the steady state assumption mimics this, and it's actually been found that this is pretty applicable to a lot of different biological phenomena. If we look at our example reaction over here, we have hydrogen and oxygen combining to form water. In steady state, we can say that stoichiometrically, the amount of hydrogen and oxygen consumed on the left hand of the equation is equal to the amount of water produced on the right hand of the equation. We can represent this mathematically in our balance equations down here. We have our products with a positive coefficient, our reactants, hydrogen and oxygen, with a negative coefficient since they're consumed, and we set the balance of this equation equal to 0. If we repeat this process for all of the reactions in our system and factor out the fluxes, we're left with a matrix called a stoichiometric matrix that represents all of the reactions present in our system that's being modeled. We're also left, the stoichiometric matrix is designated S in this equation here. We're also left with a set of unknown fluxes designated V, for which we're trying to solve. To satisfy the steady state assumption, we can set the dot product of our matrix S and our unknown vector of fluxes V equal to 0, and this sets up our system of linear equations. Additionally, we can add constraints to the individual fluxes within our system. For instance, we can set the lower bound of this flux here, water, equal to 0. This will force the production of water, thereby adding flux through the first equation. Solving for our set of equations that satisfy the steady state assumption and our additional constraints leaves us with what's called a flux cone. The flux cone is the space in multi-dimensional flux space that includes all sets of fluxes that satisfy our constraints and the steady state assumption. You'll notice that the 0 point here is also included, and this is called the trivial solution, wherein all fluxes are set equal to 0, which satisfies our constraints, but isn't necessarily biologically relevant. You might notice that this flux cone sort of feathers out from 0 and actually includes an infinite set of solutions that satisfy these constraints. Now, obviously, we can't do much with an infinite set of solutions. So what are we doing next? Well, the overall goal of FBA, flux balance analysis, is to reduce that flux space to one that is biologically relevant, that, excuse me, that mimics the actual behavior of the system as well as possible. So how do we find that optimal solution in the solution space where we have an infinite number of solutions? Well, first we can consider growth rates. We also make the assumption that if a given microbe is measured to be present in a system, it must be able to grow. If it weren't able to grow, it would die off, be excreted from the system, and we wouldn't have measured it there in the first place. So if we say that it's able to grow, in practice, this means that we can say that we want a solution in that flux cone that corresponds to maximum biomass production, since this is typically the biological goal of a growing microbe. Biomass production can also be represented as a flux, since it is, in essence, a measure of mass conversion. What's neat about flux balance analysis is that we can add information from a number of different sources in order to determine our optimal solution. As we've mentioned, we can use the reactions that are present in our system to build a stoichiometric matrix, like the one that we described in the last slide, that mathematically represents all the reactions taking place in our system and how they interact with each other. For instance, what precursors from one reaction are required in another. We can actually infer this whole stoichiometric matrix from the genome of a microbe by looking at gene abundances and then mapping those to reactions that we know are taking place in that microbe, from which we can then build a reaction network leading us to our stoichiometric matrix, which we set up as our set of linear equations. Another thing that I've already mentioned is that we assume that the system, the microbe that we're modeling, is at steady state. And so in practice, what this means is that we're modeling the microbe in the exponential growth phase. Because in this growth phase, the growth rate is constant, and therefore biomass production is constant. So we include this assumption to be able to arrive at a solution for our fluxes. We can also include thermodynamics in our solution. For instance, if we know that thermodynamically a reaction can only occur in one direction, we can set the flux of that reaction, the lower bound of the flux of that reaction to be equal to 0, forcing it to only occur in one direction, the forward direction. Finally, we can also add constraints to the system. We can also add constraints to the import of metabolites going into the system which we can leverage to represent the metabolites available to that system in order to grow. In practice, this basically allows us to model the diet that's available to the microbiome, adding relevance to the predictions that are being made by flux balance analysis. Adding all these constraints in and then solving for our fluxes, we can find that region within the flux cone that corresponds with maximal biomass production, represented by this red band here. So this is great. We now have a subset of our solution space that corresponds with the overall evolutionary goal of this microbe that we're modeling, which is to maximize its growth rate. However, you might notice that there still is an infinite set of solutions within this within the subspace of our flux cone. So the real art of FBA is to then select a single solution in that subset of solution space that's the most biologically relevant. Again, mimics the growth behavior of the microbe most closely. There's several methods available to us to find that single solution within our subspace. But today we'll focus on these last two listed here, since these are ones that are leveraged by Mycom in order to find a solution. One such solution is to minimize the imports into a system. Since the microbiome can be a highly competitive environment, this is biologically intuitive, since the most successful residents of the microbiome will not be relying on importing lots and lots of different metabolites in order to survive. And rather, they're able to subsist on a smaller subset. Another common approach is parsimonious flux balance analysis, sometimes called PFBA, which basically minimizes the number of enzymes required to satisfy the constraints of our flux balance analysis. PFBA has actually been found to reproduce experimental fluxes in E. coli very well. And intuitively, this makes a lot of sense, since bacteria are very efficient beings that don't like to produce more enzyme than they need to in order to grow. So really, the method that you should use in order to find your solution in your reduced subspace is related to the question that is being asked and the system being modeled. For instance, is it competitive? Is it resource limiting, etc.? I encourage you to look more into these methods if you're interested in conducting your own flux balance analysis in the future. So to this point, we've been focused on how flux balance analysis can be leveraged in order to simulate genome scale metabolic models. So how we can determine the fluxes in a single bacterium. To make metabolic models of microbial communities or metagenome scale metabolic models, we use a tool called Mycom, which extends flux balance analysis into microbial communities. So to first build these models, we need to pass in the relative abundance of the bacteria in our sample. Due to differences in sequencing efficiency, there may be some bias towards some bacteria over others, but the abundance from our sequencing data should be more or less representative of the community that's being modeled. Mycom will then map the abundances of these, will then map the abundance data to a database containing hundreds of genome scale metabolic models for common gut microbes. So basically a database that contains hundreds of reconstructions of individual taxa that are commonly found in the gut microbiome. Mycom then uses these reconstructions of the taxa that are present in the sample to build a massive stoichiometric matrix like the one that we described earlier, that includes not only the internal reactions within each taxon, but also exchanges between taxa and external reactions, basically imports and exports out of the community to the external environment, which in this case is the gut lumen of the host. After this, we can then specify particular diets that basically indicate what metabolites are available to the microbiome in order to grow. Mycom will then use FBA with an additional regularization step that we'll talk about in upcoming slides in order to calculate unique growth rates for most of the taxa present in our community. Using these growth rates, Mycom then estimates fluxes that are occurring within this community and returns those as predictions. So all in all, this gives us a huge amount of output data that we can use that we can then use to make detailed and interesting predictions about imports and exports into and out of our microbial community. It also tells us about the sort of inner network of reactions occurring within each bacteria that it's typically very difficult to measure. It can tell us about codependencies between bacteria. So for instance, if one bacteria is dependent on metabolites produced by another or inhibited by metabolites from another, we can model that in these metagenome scale metabolic models. And it also provides us with sort of a testing ground for potential interventions. So if we want to see how different dietary contexts affect the metabolic output of the microbiome, we can do that with these metagenome scale metabolic models in Mycom. So here's a quick overview of sort of the typical workflow within Mycom. Like I mentioned previously, we start with feeding that abundance data in, as well as a database of genome scale metabolic models. Mycom then maps the abundance data to that database and pulls in reconstructions of all the taxa present in our model. We then apply a medium and allow Mycom to predict unique growth rates for most of the taxa present in our community, and then also predict fluxes based on those growth rates that are then output to the user. We can then use those predictions for growth rate and exchange fluxes in order to build visualizations and interpret our results. What's really neat about Mycom is that it was built to be supported by two separate interfaces. There's the Python interface that we'll be using today in our Google Colab notebook, but you can also run all of these commands directly in QIIME 2. So if you start your pipeline in QIIME 2 and would like to run the whole Mycom pipeline there, you're able to do that. What's also great is that it's very easy to switch back and forth between the two interfaces. So if you start your Mycom build process in QIIME 2, you can then take those QIIME artifacts that are produced and bring those into the Python interface and continue the pipeline there. So this is a really great tool that's very extensible and easy to use. Great, so now that we have a basic idea of flux balance analysis and a quick overview of Mycom, let's switch back over to our notebook and we can take a look at how we'll go about building and growing our communities. Right, so all of the setup is now complete for our notebook here, and we can jump into actually using Mycom. So again, here's just a quick overview of the Mycom pipeline, if you'd like to familiarize yourself a little bit more. And like I mentioned in the slides, the process of building metabolic models in Mycom begins with constructing a combined abundance taxonomy table that after this point, we'll just refer to as a taxonomy table for simplicity. Before we load in our own data, we can import an example taxonomy table directly from Mycom and take a look at what that should look like so we can compare that to our data that we constructed yesterday in QIIME 2. Let's run this cell and take a look at the output. Cool, so this is what a typical taxonomy table for use in Mycom should look like. You'll notice in this table, we have 4 identical strains of E. coli spread across 2 different samples, sample 1 and sample 2. We can see that each row here represents a single taxon within a single sample, and that we have a few different columns here, each of which are important. So there are 4 required columns in a taxonomy table in Mycom. First, we need this ID column, which basically specifies the unique identifier for each taxa in our community models that we're building. We then also need a column specifying the lowest rank to which we're building the lowest taxonomic rank to which we're building our community models. In this case, we see that we have the species rank, indicating that we're building models at the species level. Mycom will use the name of the species here in order to map to that model database that contains all of the individual reconstructions of taxa that are being used to build the metagenome scale metabolic models. If we include higher order taxonomic ranks, like for instance here we have genus, Mycom will use those in order to map to the database, but they aren't required. So any additional taxonomic information you pass in will be used in order to map to the database. But again, it's optional. And in fact, if your database and your taxonomy table are annotated differently, this can lead to situations where maybe a taxon is named differently in the database than you have in your taxonomy table and is then omitted from your communities as a result. So just be very careful about how you're naming your communities in your taxonomy table and make sure it's consistent with the database that you're using. We also see that we have a sample ID column. This is required to tell Mycom which sample each of these taxa is residing in. And then we have an abundance column, which in this case shows us read counts and just tells us how abundant that taxon is in that given sample. Cool. So you might notice that, yesterday we built tables in Chime. And so that's not necessarily so easy to read in, but no worries, we can deal with that. Like I mentioned previously, Mycom is built in order to be able to accept Chime outputs. And that's why we installed those two packages early on, allowing us to take a look at Chime data directly in Mycom. So we will import our data here shortly, but before we do, I'd like to touch on that model database that I've been alluding to a few times throughout this course. So basically what we need in order to build our Mycom models is a database that has pre-curated reconstructions of individual taxa. And then Mycom will scrape that database for the taxa that are present in each one of our samples and use those individual reconstructions in order to construct our metagenome scale metabolic model. So we'll be using the Agora database, which is a curated database of more than 800 bacterial strains maintained by the Thiele lab. And these are strains that commonly live in the human gut. In particular, we'll be using a version of this database summarized at the genus rank, since we will be building our models at the genus level, since that is the lowest taxonomic rank that is we can get reliable information from 16S data, like Christian mentioned yesterday. This Agora database can be downloaded from the Mycom data repository if you're interested in using it further. But we also copied that over from our GitHub repository earlier, so we don't need to worry about downloading that ourselves. It should be in our materials folder on the left-hand side in your file explorer. You see it right here, Agora 103_genus, and that's also a Chime artifact. Great, so now we have that out of the way, we can get into loading up our own data and building our taxonomy table that we'll use in order to build our communities. So to do this, we'll import this Chime to Mycom function from Mycom, which will allow Mycom to read in our Chime artifacts, specifically our table artifact, which contains abundances, and our taxa artifact, which contains our taxonomic mapping. We'll also include this collapse on argument, which summarizes the abundance data to the genus level. Since again, we're building our models on the genus level. So we'll run this and this will go pretty quickly. And again, we only had to specify the genus level since that was what we were using to build our, since that's the level that we're building our models for. Great, so we can now take a look at the output from our Chime to Mycom function and take a look at the taxonomy table that was generated. Awesome. So this looks a lot like the example table that we looked at just a second ago. We have our sample ID column, which specifies the individual sample that a taxa is present in. We have an abundance column telling us the read count of that of that individual taxa in that sample. We have our lowest taxonomic rank summarized in this table here. We see that we're using the genus level, so we have individual genera names here. We also have an ID column, which again is a unique identifier for each one of these genera. However, you'll notice that in this case, the genus column and the ID column are identical, and that's okay. It doesn't have to be, but that works just fine. We also have a relative abundance column here, which basically just gives us abundance again, but this time in compositional form as relative abundance rather than absolute read count. Awesome. So now that we have these, our taxonomy table ready to go, we're theoretically ready to build our models. But before we do that, we'll do one more thing, which is merging our taxonomy table with our metadata. So if you recall yesterday, we had metadata available for each one of our samples. We will merge these tables together, and that way we'll have it on hand for following steps. So we can always go back and reference the metadata in upcoming steps. So if we run this and then take a look at our output, we see that we still have our same taxonomy table, but now we've merged it with our metadata. So in addition to abundance, we also have ethnic group, subsistence type, and country of origin for all of our samples. Cool. So with our taxonomy table ready to go, and now our metadata is merged, now we are really ready ready to start building our models in Mycom. Cool. So now that we've really set ourselves up nicely, building the models is fairly straightforward and really should be a breeze. So in order to do this, what we will do is import this build command from the Mycom workflows module. And then in this build command, we'll start running this now, and then I'll talk through a little bit of what's happening in the background of this build command and some of the inputs that we put into it. So in this build function, we'll pass in tax, which again is our taxonomy table that we just built from our CHIME artifacts we constructed yesterday. We'll pass in the name of our model database. So again, this is the Agora database containing genome scale reconstructions of several hundred common gut microbes, excuse me. We'll also pass in the name of the model folder that we'd like to have our model saved to. So as Mycom goes through and builds individual models, it will save these models in a serialized form in the output folder that we specify here. So you'll see that now on the left-hand side in our file explorer, we now have an output folder called models, and that's where all of our models will live. We'll specify the solver we're using, which again is OSQP. If you were to get an academic license of a different solver, then you could change the solver that you're using, and you might get better numerical efficiency. This cutoff command here specifies the relative abundance threshold that we'll use in order to build our models. So in this case, our cutoff is 0.01, which designates 1% relative abundance. And so what that means is that any taxon present in our sample at less than 1% relative abundance will be omitted from the models. Now we do this in order to, since we assume that taxa present in that low abundance aren't substantially contributing to the metabolic output of the models. I do urge you, if you are planning to use Mycom on your own, to take a look at that cutoff and determine what is a good cutoff for your own analyses. Typically, we'll use a slightly less strict cutoff, but for the purpose of this course and to minimize the sort of computational intensity that was required, we elected to go with a 1% relative abundance cutoff, and that seems to work just fine. We'll also parallelize this process over 2 threads, which is what this command does here, and that'll minimize the amount of time that's required in order to run this command. Great, so this all in all should take about 10 minutes to run, and so while that's going, we'll switch back over to our slides, and we'll discuss a little bit more about the next step in the Mycom process, which again is is growing the models that we're building here. Great, so now that we've covered like classic flux balance analysis that we use for genome scale metabolic models, so I think the metabolism of one single microbe, everything seems pretty straightforward. You just maximize the growth rate, you find that maximal biomass subspace within your solution space, and you get your answer for your fluxes. So can't we just do the same thing for community models? And the answer is not quite. So in a genome scale metabolic model, for a single bacterium, we only have one growth rate, which we can designate mu. In a microbial community, however, we have several growth rates. So we have mu sub I for each individual growth rate, for each individual taxon in our community. And we also have an overall community growth rate designated mu sub C. So mu sub C can be calculated by summing together the abundance of each individual taxon multiplied by its own individual growth rate. So could we just maximize the community growth rate and then get an answer that way? Well, not quite. If we try to just maximize community growth rate, you'll find that we pretty quickly run into trouble. And we'll discuss why right now. So in practice, getting a community-wide growth rate using just classic flux balance analysis doesn't work very well. Let's set up an example here to illustrate. where we have two identical microbes in our community. We can say they're both E. coli, and they're completely identical, except for their different colors. And let's say we have them in the exact same abundance, so 50% relative abundance for each. If we know that the maximal community growth rate is 1, we can see that there's actually an infinite number of solutions to this. We could have both of them growing at a growth rate of 1, which after we multiply by relative abundance, still comes out to a community growth rate of 1, based on our definition in the last slide. But alternatively, we could also have a growth rate of 2 for this blue E. coli, while the orange E. coli is not growing at all. This would still give us a community growth rate of 1, despite the fact that 50% of the taxa present in our community aren't growing at all. So this plot here actually shows us this. We have this red line representing our optimal maximal community growth rate, and we can see that there's an infinite number of solutions on this line that satisfy that maximal community growth rate, including these terminal solutions here, wherein one of our taxa is growing at a high growth rate, while the other one is not growing at all. So this isn't good, since this introduces a lot of ambiguity to our system, and it increases the size of the flux cone to include non-biologically relevant solutions. And that's obviously not what we want. Intuitively, we know that if we have two identical strains, they should be growing at about the same growth rate. And if we make the assumption that we mentioned earlier, which is biologically intuitive, that any taxon present in a sample should be able to grow, we shouldn't include these solutions wherein one taxon is growing and the other isn't. So to overcome this, Mycom introduces a regularization technique that reduces our solution space back to one that's relevant. So again, if we make that assumption that anything present in the sample should be able to grow, the regularization strategy should return us a solution in which most taxa are growing, omitting those solutions wherein one taxa is growing at a fast rate and the other one is not growing at all. What's called an L2 regularization scheme actually achieves this pretty well. And it does this by minimizing the sum of the squares of the individual growth rates. Basically, what this does is penalize those solutions wherein one growth rate is really high, and it distributes growth back across as many taxa in the community as possible. And indeed, we see that if we apply this L2 regularization scheme to our example community here with our two identical E. coli, it finds an optimal solution wherein both taxa are growing at the exact same growth rate, with a growth rate of 1. And in fact, L2 regularization will always find a unique solution along that line of maximal community growth rate that tries to distribute growth as evenly as possible across taxa. You might, though, if we approach this biologically, though, it does make sense that for all the bacteria to be able to grow in a community, optimal growth might not actually be able to be achieved. An idealistic maximal growth rate might only occur when some really fast-growing bacteria are growing at the expense of other slower-growing bacteria. So in Mycom, we can model this by incorporating a cooperative trade-off parameter. And basically what this does is specify the fraction of the maximal community growth rate to which we want to perform our L2 regularization. So for instance here, if we set a trade-off parameter of 0.8, so 80% of maximal community growth rate, we see that our L2 regularization finds that optimal solution at 80% of the maximal community growth rate, lowering our growth rate slightly. And you'll see in the next slide why this is sometimes more biologically relevant than that optimal solution along that maximal community growth rate line. Methods for choosing a trade-off parameter are covered at the end of the course notebook. So if you're interested in performing your own analysis, Mycom, I urge you to take a look at that after the course is complete. So cooperative trade-off flux balance analysis, like we just covered, allows us to treat those metagenome scale metabolic models that we're building with some of the same methods that we do for genome scale metabolic models. So again, we can use parsimonious flux balance analysis, for instance, or minimization of imports in order to find a unique solution within our flux space. But the age-old question, does it actually work? Does cooperative trade-off flux balance analysis actually give us answers that are biologically relevant? So here on the left, we can see the results from a sample Mycom community with the number of taxa growing at various trade-off values. For reference, a growth rate less than 10 to the minus 6 is effectively no growth at all. So we consider this gray shaded region to be taxa that are not growing in the community. We see that in this community, when it was grown without an L2 regularization scheme, we see a very small number of taxa able to grow at an appreciable rate, with almost all of the taxa present in the sample not growing at all. We then see that if we do apply our L2 regularization scheme, still optimizing to that maximal community growth rate, we see distribution of growth across many more taxa. So we see many more taxa that are able to grow in that region to the right of the gray shaded region. We still see some taxa that are not able to grow, but growth is distributed much more evenly across the taxa. We then see that lowering that trade-off value to about 0.7, so performing our regularization strategy to about 70% of the maximal community growth rate, we see that almost all of the taxa are able to grow, with only a few taxa not able to grow. So this actually already satisfies the assumption that we made previously, which is that taxa that are present in the sample should be able to grow. And this sort of echoes what we're expecting to see in an actual microbiome, which is that most of the taxa are growing. So this in and of itself is already an indication that the L2 regularization scheme with cooperative trade-off seems to be giving us more biologically relevant results. The next thing the team wanted to answer, though, is are those growth rates that are being predicted by Mycom actually relevant to what we see in the communities? So for these data that they were using, the team had metagenomes, and it's been shown by the Segal lab that coverage profiles of metagenomic reads, like the one seen here, can be used in order to estimate a growth rate. You can do this by measuring the decay and read coverage from the origin of replication as it moves away, and from there infer a growth rate in fast-growing taxa. So using this method, the team could actually measure growth rates in vitro using this replication rate estimation method and compare those to the growth rates that were predicted by Mycom. So what we see is that without the L2 regularization scheme, or with the L2 regularization scheme, but still maintaining that maximal community growth rate, there was very little correlation between actual growth rates measured in vitro and those returned by Mycom. Again, the team saw, though, that bumping down that trade-off value, so optimizing to 70% of our maximal community growth rate, actually returned growth rates that were much more correlated with what was seen in vitro. So really what this tells us is that cooperative trade-off flux balance analysis satisfies our assumption that taxa that are present in the community should be able to grow, and it returns to us growth rates that are actually biologically relevant as compared to other methods without L2 regularization. This also shows us that optimizing to that maximal community growth rate, even with L2 regularization, isn't always the correct technique, and that sometimes sort of a suboptimal growth rate is the one that's actually the most biologically relevant. So that trade-off value is actually very important moving forward as we build and grow our models in Mycom. Cool. So all these methods seem great, and all the math seems to check out. So what's taking so long to run all these workflows? Well, typical Mycom models, like the ones that we're building here today, are actually a bit more complex than the examples that I've been using to illustrate the methods, like, for instance, our 2 E. coli community. And actually, we can take a look at that here. So this is a visualization of a standard metagenome scale metabolic model that you might build in Mycom. So each pink dot in the visualization here represents a single metabolite, and each blue dot represents a reaction. And those reactions and metabolites are connected with black lines. you'll see that each of these regions that seems to be a little bit distinct from the others represents one taxon. And there are thousands of internal reactions as well as exports and exchanges between individual taxa and with the outside environment. So you can see that this community model shown here has nearly 70,000 reactions, over 45,000 metabolites, and nearly 300,000 unique connections between those. So you can imagine that finding an optimal flux solution through this model takes a little bit of computational power and sometimes a little bit of patience. So it can take a little while to run these models, but the output from Mycom gives us an immense amount of data that that finds sort of the most biologically relevant flux through this massive model that's shown here. Awesome. So now that we have a better understanding of some of the methods that Mycom and assumptions that Mycom makes during the modeling process, we can switch back over to the notebook that we've been using. The model should have built at this point, and we can move forward with the next step in the process, which will be growing our models, and from that, developing an output that we can use to build visualizations of our results. So we'll switch over to the notebook. And hopefully at this point, your models have completed building. If not, please go to the Slack and the TAs will be able to help you there. Before we move past this cell that we just ran, I want to call your attention to this warning that was given underneath the cell that we just ran, wherein we built our models. So you'll see this warning that says less than 50% of the abundance could be matched to the model database. Model Hadza3 may not be representative of the sample. So what this means is that Mycom took the taxa present in this model and tried to map them to the model database, the Agora database we're using, and it wasn't able to find reconstructions for more than 50% of the abundance of the taxa in that model. Typically, this would be some cause for concern. Typically, a fraction of 80% or more is considered great, and we really shoot for greater than 50% abundance matched to the model database. However, in this case, this is one of the issues that we face when modeling underrepresented groups in microbiome research. The database that we're using, in this case, the Agora database, has been built based on microbiome studies of overrepresented groups, namely European and North Americans. So updating these databases to be representative, not only of those individuals, but also of these underrepresented groups, like the ones we're modeling today, is one of the things that the microbiome research community needs to address in upcoming studies in order to make microbiome research more equitable and applicable to individuals all around the world, and not just in those overrepresented groups in North America and Europe. All right, so with that out of the way, let's take a look at the output from our build command. I do want to mention that we will continue with this model, even though less than 50% of the abundance could be matched. But it is an important thing to keep an eye out in future projects. Again, it is something that the microbiome community needs to address in coming years. So the output of the build command gives us this file that we've named manifest. And so what we see here is, again, our sample IDs, as well as the metadata that we've passed along into the build command. And on the right side of this data frame, we see our statistics from the build command. So this found taxa column designates how many of the taxa in the sample were mapped to our database. The total taxa is just the total number of taxa that were present in the taxonomy table that we passed into the build command. The found fraction is very easily just the found taxa divided by total taxa. And then the found abundance fraction is that found fraction weighted by relative abundance of taxa in each one of these samples. So we can see that most of these seem to be above 70% at least, which is pretty good. And then we see a few like this one here that's below 50% that don't have quite as many taxa mapping to the model database. And that's all right, we'll continue with that. But that's a good thing to keep an eye on when you're building your own Mycom models. Great. So now we have our community models built, and now we can leverage Mycom to conduct the next step, which is simulating community growth. But before we jump into that, let's discuss a little bit what it is we want to look at with our Mycom models. So let's think a little bit about the insights that we might want to gain from these samples. So first and foremost, we can use Mycom in order to investigate the metabolomic response of the gut microbiome. And we've discussed that a little bit. And one thing we're interested in is how that metabolic output changes based on dietary input. So we can use Mycom to take a mechanistic look at how the dietary input changes the metabolic behavior of the microbiome. So for instance, if you were to take individuals from an indigenous population, like the ones that we've been modeling here today, and we were to simulate the growth of these models on a matched diet, so a diet that's representative of a typical meal eaten by an individual from that population, and then simulate it again on an unmatched diet. For instance, today we'll be using a representative Austrian diet. How does the metabolomic profile of the microbiome that we're modeling shift? What exports are different? How does the growth behavior of each of the individual taxa change based on the dietary input that we feed the microbiome? So that's sort of the overview of what we plan to do with our models here today. So as I mentioned previously, before we can conduct the growth simulation, we need to pass in a medium that designates which metabolites are available to the microbiome in order to grow. So to do that, first we need to load our medium. So in this cell, we'll download the matched diets for each of the 3 communities that we're studying that was constructed by our team by hand. This is a CSV file, so we can easily read it using pandas. So we'll run this cell. And if we actually take a look at the output from this, just type this, and we look at the output, we'll see that this data frame is pretty long. And that's because the medium shown here is per sample, which means that each of the 9 models that we've built in the previous step has its own medium represented in this file, which is dependent on the food typically eaten in the community that they live in. So we have actually 9 separate media represented in this file, along with sample IDs, so they will be applied only to those taxa, only to those samples that they correspond with. Great. So now that we have our media loaded and we have our models built, it's time to actually grow our models. So again, we have 9 individual samples that we're using, and we have this per sample matched medium. So we've actually set ourselves up very well, and this grow command will be, again, pretty straightforward and easy to use, but it will take some time. So I'll start running this cell here, and I encourage you to do the same. And then we'll discuss a little bit about what's going into this command. So again, from the Mycom workflows module, we'll import our grow command, as well as our save results command. We'll then run this grow command, which will take the models that we've built and it will apply cooperative trade-off flux balance analysis in order to estimate unique fluxes for most of the taxa present in our community. And from there, also estimate fluxes happening in each of these taxa, and report those back out to us. So in grow, we will pass in our manifest that we got out of the build command previously. We'll specify the folder that we saved our models to. So if you recall, we saved all of the models in serialized form in this models folder shown on the left-hand side in your file explorer. We'll designate the medium that we want to use, which again is this matched medium, which is representative of a typical meal in each one of the indigenous communities that we're modeling today. And we'll also specify a trade-off value, which, if you recall, is the fraction of the maximal community growth rate to which we're running our L2 regularization scheme. In this case, we'll use a trade-off of 0.5. This seems to give us pretty good results, but if you are to run your own Mycom models on your own time, I do encourage you to take a look at the end of this notebook at the trade-off analysis section, which will help you understand a little bit better how we go about selecting a trade-off value in Mycom and moving forward with that. So we'll parallelize our process here across two separate threads, which will increase the power or the computational speed. And then once that's all done, we'll save our results using the save command, passing in the results of our growth function, and then specifying where we'd like to save our results to. So in this case, we'll save a zip file of the growth results in our treasure chest. If at any point, if we come back to this and it hasn't completed for you, or if for any reason this cell was aborted, feel free to read in the results from the treasure chest. They already exist there, and it should match exactly what sort of output you would be getting from this. So we'll let this run. All in all, this should take about 20 minutes. But yeah, with that, I think now would maybe be a good time to take some questions from the Slack, if there's any available.
So there were a lot of questions about model databases, especially when you wanted to model maybe factors from other body types, like vaginal microbiome or oral microbiome, and also like mouse that, like an overview.
Sure. Yeah, so the question was about model databases and what sort of databases might be available for the microbiome, but also for modeling other body sites, perhaps, or in other in the mouse gut, for instance. Off the top of my head, I'm not very familiar with many other databases, but I do know that there are many curated databases available on the web that you could find and pass into Mycom. I think the Mycom data repository has a few databases including the Agora database at a few different summary ranks. So if you were to build the models of the microbiome at sort of the family rank or at other taxonomic ranks, you can pull in the Agora database from the Mycom data repository there.
There's also some questions about the limitations of FBA and that it can't account for direct toxicity, like direct killing, bacteriocin, or toxicity from tell. It only accounts for metabolic interactions. Talk about that.
Yeah, right. So the question was about the limits of FBA, of flux balance analysis, and whether it's able to account for toxicity of heavy metals and other interactions. And yeah, so flux balance analysis really is limited to sort of metabolic interactions. And doesn't really account for many of those things. For instance, one difficult thing to model is pH in the gut, and that's something that isn't fully captured by flux balance analysis. And there are proxy ways to sort of model these things and include them in the community model. But flux balance analysis itself does have limitations in the sense that it really is limited primarily to metabolic interactions. But that is the case, yeah. All right, well, yeah, I'll jump back into the slides for a little bit here, and then we'll continue. Yeah. So while our communities while our models are building, let's discuss a little bit about, we'll revisit the theme of this year's course, which is, again, modeling the microbiome of historically underrepresented groups within microbiome research. So this visual, which Chris showed us yesterday, shows us just how overrepresented the North American and European microbiomes have been in the field of microbiome research in the past decade. Today, we will build metabolic models of the microbiome for the 3 indigenous groups introduced in yesterday's lesson, which we've already started. And we'll use these models to make predictions of the metabolic behavior of each based on the environmental context that the microbiome is exposed to. We'll also see what work still needs to be done in order to make microbiome research more inclusive of these historically underrepresented groups. And we'll see how current methods can be shaped by overrepresentation of subsets of the population, such as European and North American microbiomes. So as I've already touched on, for instance, the model database that we used to build our models was constructed primarily based on studies of European and North American microbiomes, and therefore is overrepresented by taxa commonly found in microbiomes from those regions. All right, so when I say the environmental context, what do I mean by this? This is the environmental context that the microbiome is exposed to is a key determinant of composition and subsequent metabolic response. So this can take multiple forms. For instance, we have different water sources can impact the composition of the microbiome and subsequent metabolic behavior of the microbiome. Drug use can, for instance, antibiotic use can drastically change the composition of the microbiome and cause large shifts in composition and then ultimately in the growth behavior of the microbiome. Exposure to pets and other animals can introduce new microbes into the microbiome and again, change its composition and therefore its metabolic behavior. And then finally, diet is one of the most important contributors to this environmental context and can drastically change both the composition and the metabolic behavior of the microbiome. So in Mycom, as you've already seen, we can partially represent the environment we can represent the environmental context through the application of an in silico medium that represents the diet available to the microbiome. Since metabolites in the diet are directly feed into internal reactions and are direct precursors of microbially produced metabolites, it's critical that the medium that we use for the growth simulation within Mycom be representative of the diet that's typically consumed by the subject being modeled. If we were to apply a diet that doesn't represent what that individual is actually eating, the predictions that we gain from Mycom will likely be very inaccurate. So today we'll be investigating this by using two separate media for the samples that we've built in Mycom. First, we'll be using a matched medium, which represents the diet of the indigenous groups that we are modeling. And secondly, we'll be using an unmatched medium. In this case, again, we'll be using a representative Austrian diet, which is not typically consumed by the individuals in these three indigenous groups that we're modeling today. Okay, so to use a medium for a growth simulation in Mycom, the medium must be componentized. So what does that mean? That means it needs to be described by its constituent metabolic components. We can't yet say that this person just ate a banana. We rather have to determine what metabolites are in the banana and then add those metabolites to the medium individually. Componentizing media itself is a pretty laborious task, and it requires determining the constituents of various food components present in the diet and then adding them in the proper amounts. For diets of highly represented groups in microbiome research, such as North American or European diets, componentized media are publicly available and easy to find online. However, media representative of the diet of underrepresented groups, like the three indigenous populations we're studying today, is not commonly available and thus needs to be constructed by hand. This can be additionally difficult since the databases that you might use in order to build these media by mapping food components to their constituent metabolites, those databases consist primarily of foods that are common in North American and European diets. For instance, baobab fruit, which is shown in the picture here, is a very common component of the Hadza diet, but it can't be found in any of the commonly used food databases that we use to map food components to their constituent metabolites. The construction of media that's representative of the diet of underrepresented groups rather than the diet of European and North American individuals, as well as the inclusion of food consumed by these groups in these food databases that we're using to build the media is another issue that must be addressed by the microbiome research community in coming years to make this make research in this field more representative of the world population and more applicable to the world population, rather than just those groups that are historically overrepresented, again being European and North American microbiomes. So for this portion of the course, we decided to construct our own media representative of a standard representative of a standard meal in each of the 3 communities that we are modeling. So we built a componentized representative meal for the Hadza, Chepang, or Mfa communities, each of which were very distinct from each other. First, the first step here was to determine what the meal looked like based on extensive literature review. So for the Hadza community in Tanzania, a representative meal included baobab fruit and seeds, honey, and antelope. For the Chepang community in Nepal, we included nettles, mountain yam, and cowpea. And finally, a representative meal for the Mfa community in Mexico, corn, beans, chayote, and chicken were included in the meal. Each of these is representative of a meal in each of these communities, but is not by any means an exhaustive representation of their entire diet, which would require a much more extensive investigation. Additionally, seasonal dietary variations introduce shifts in metabolic availability to the microbiome that is not captured here. However, each of these diets is a much closer approximation to much closer match to the corresponding community upon whose diet was based than the existing componentized media that we could find online. And the metabolic components in each were quite distinct from the unmatched representative Austrian diet that we're using as our unmatched medium. In order to construct the matched media that we'll be using for modeling the communities, we first had to determine the dietary components present in the standard diet of each community, as I explained in the last slide. So we basically found the food components that are present in a typical meal for each one of these communities. Next, we mapped each food to its constituent components using a food database. Like I mentioned previously, some foods like baobab fruit were not available on these databases. So for instance, in the case of baobab fruit, we found an NMR-based study detailing its metabolic composition, which we could then use to determine those constituent metabolites and their abundance. After determining these constituent metabolites, we add those to our medium in the correct relative abundance. And then following this, we then have to account for adsorption in the small intestine. So many of the metabolic components of our diets are actually absorbed in the small intestine before they even make it to the gut microbiome in order to be consumed there. So we have to account for that in this step. We also add bile acids and mucin, which are introduced along the gastrointestinal tract before reaching the large intestine. And then finally, before we have a completed medium that we can use for growth in Mycom, the medium undergoes a gap filling process, basically augmenting our medium with a minimal set of metabolites that are required for all of the taxa in the sample to be able to grow. So now at this point, we have a componentized medium for each one of our 3 indigenous communities that are being studied. And we can apply this to the models that we're growing. And with cooperative trade-off FBA, we can then use those to return unique growth rates for most of the taxa present in our sample, and then estimate fluxes from those growth rates. Excuse me. All right, so I think at this point, our models are likely still growing. So this might be a good point to take a break and then come back to the presentation after the break. And we'll, after the models are finished growing, I encourage you to leave your notebook open to let those complete growing. And then we'll take a look at the output of our results and build some visualizations based on that output. So thank you so much for your attention.